Welcome to Swastome, modern solutions for healthcare, built for India's tomorrow, shaping the future today.
My great pleasure to introduce our next speaker, Dr. Ayushi Verma, a distinguished project research scientist at the ICMR Center for Advanced Research in Aging, where she contributes to cutting-edge research on the biology of aging as part of multidisciplinary efforts in longevity science. Dr. Verma is a clinical omics researcher with a PhD from Indian Institute of Technology, Bombay, and her work exemplifies the convergence of high-throughput molecular science and clinical research. She specializes in integrative genomics, proteomics, and metabolomics, applying these multi-omics strategies to decode complex disease pathobiology and translate large-scale molecular data into actionable insights. Her research focuses on biomarker discovery, pathway analysis, and advancing precision medicine, areas critical for understanding disease mechanisms and developing tailored therapeutic approaches. Dr. Verma's expertise in analyzing high-dimensional clinical datasets not only enhances our understanding of human health and disease, but also paves the way for innovations in personalized diagnostics and treatment strategies. Please join me in welcoming Dr. Ayushi Verma to the stage.
Yeah, let's talk about the role of proteogenomics in cancer research. We know that from DNA to protein, information carry forwards to the protein level from DNA, and cancer starts at the genome. A lot of time mutation happens at the DNA level, and that mutation may or may not get translated at the protein level, right? So we need to understand that not all the time the mutation that is happening at the DNA level will be translated, and which mutations are getting translated at the protein level will lead to the mutated peptide formation. And cancer can be due to a lot of factors are involved that changes the DNA and ultimately lead to the cancer. One is the if mother or father has history of cancer, their progeny is more they have more risk towards the cancer. UV radiation, physical, chemical exposure, viruses, smoking, abrupt division of cells, all these can lead to the cancer. And the change in DNA can have different kinds of mutations on the DNA, copy number changes, gene fusion, epigenetic alterations, all that. But we need to understand that not all mutations produce the protein, get translated at the protein level. So genomics is definitely not enough if we want to understand what is happening at the protein level. But hope I'm audible. Yeah.
Yes.
Okay. So a lot of big consortiums are established, like the Cancer Genome Atlas, where thousands of tumors are sequenced. They have catalog driver genes and all the mutations which are present in those cancer, like breast cancer, cervical cancer, brain tumor, glioblastoma, one of the brain tumor. So all those databases have huge genomic information, but genomics will only tell you what could happen, and that thing might not necessarily happen, right? Those genes might not necessarily get translated at the protein level. So that is the one factor that we need to keep in mind. Another thing is earlier, so I'm giving you one example of brain tumor where in 2021 WHO has incorporated a lot of genomics marker along with the histology. So earlier classification before 2021, only the histology based markers are there for classify the adult diffuse gliomas into different subtypes. But after 2021, they have included various genomics marker like IDH, which is isocitrate dehydrogenase, ATRX, TERT, EGFR, all those mutations are now getting considered if we want to segregate the tumor better. So why this subtyping classification is important? Because if you subtype them, you will be able to give them precision treatment, right? You will be able to give, because one type of treatment will not work for all those subtypes. So some mutation is overexpressed, you can target that particular mutation for the therapy, right? So that's why first step towards precision medicine is first you subtype them. So that's why genomics have now been included in WHO classification of brain tumors, and we are hoping that they will include the next classification. They'll include proteomics markers as well. So now genomics will, earlier genomics was working very fine, but then while there's a need for proteomics. So in the proteomics, they will tell you that, as I was saying, that not all the genes which are present in the DNA will get translated at the protein level, and you need to understand what are the proteins which are actually doing the job in the cancer. What are the proteins which are getting expressed in your cancer sample? So for that, proteomics is the way where you take the sample, tumor sample, compare it, and you also get the healthy sample so that you know those proteins which are expressed in your tumor sample versus healthy sample. So you do the mass spectrometry analysis, you identify the certain proteomic signature, right? And those signature can be a therapeutic use, like they can be used in the diagnostic, prognostic. So you can detect a lot of protein level information using proteomics. So of course you will get the abundance value, where you will see, okay, this particular protein is highly abundant in my tumor sample compared to the healthy sample. Another important factor is post-translational modification. So sometimes you will see your proteins are not getting changed, right? So when you compare the expression, basal level expression of protein, they are same as the healthy, but maybe they are getting modified. Maybe some phosphomodification, glycomodification is happening, and their folding is getting changed. One very good example is the tau protein in neurodegenerative diseases, where basal level expression did not change, but there, because of the phosphomodification, their folding changes, and then they start aggregating and then lead to the neurodegenerative disease. So all these information you can only get by doing the proteomics. You won't be able to get all these information with genomics, right? So now, why, if the proteomics was giving you all information, then why there's a need for proteogenomics? So proteogenomics is the thing where you integrate genomics and proteomics to understand how genomic translation alterations are affecting the protein expression and function. So one very good example, when you identify the protein, right? So those protein, when you get the proteomics data, you fragment the protein using trypsin enzyme, right? And then those peptides will be identified by the mass spec, and those peptides will again map to the in silico databases, and then you will identify the protein. So basically, mass spec is identifying peptides, not proteins. So it will identify the peptides, and then by using the database, you will map those peptides to the protein, right? So the role of database is very important in proteomics. So if you are only using proteomics, you will be working with the normal database, like healthy person databases, right? So that database is not enough. So for the precision therapeutics, you need to perform the genomics of the patient, of the patient, and then create a customized database. Basically, what proteins are actually there in the patient, and then customized database use to map your proteins. To identify the proteins at the proteomics level. So this customization is very important for the precision medicine, or the custom, if we talk about the patient specific therapy. So you need to perform the genomics to create the customized database, and then this database you use for protein identification. That will give you the actually getting the proteins that are getting altered at that particular patient, because if you're using the normal databases, you won't be able to identify those hits which are actually important for that particular patient, right? So because tumor heterogeneity is there, patient heterogeneity is there. So using only the standard database is not enough. So that's why proteogenomics is a tool where you can combine the very powerful technologies, genomics and proteomics, and then you predict the you can identify the biomarkers or the mutant peptides or the novel pathways, right? And in those lines, global initiatives are working. One of the very important initiatives is CPTAC, which is the Clinical Proteomic Tumor Analysis Consortium. They usually publish very huge data sets of proteogenomics, multi-omic study, where they dissect different kinds of tumors. right? So we need to understand that, let's say, just to give you an example, that there are 1,000 mutations at the DNA level, right? Those 1,000, out of those 1,000, only the 400 are getting at the transcript level, and out of those 400, only 50 are actually reflecting at the protein or peptide level. So proteogenomics will help you find those 50 important mutant peptides or translational functionalities, which will be lost within these 1,000 mutations if you only look at the genomics data. So of course the importance, there are various kind of importance, and um, earlier I was talking about the subtyping and classification. So this slide represents how the various omics technologies are getting included with each year to increase the subtyping or classification of tumors. So earlier only the microarrays were the thing where they were used to classify the tumors or subtyping the tumor. With the genomics, high throughput genomics or sequencing coming in, those were included in subtyping, and now the proteomics, multiomics are the thing where proteomics marker, maybe the metabolomics marker will be included in the future for better classification and subtyping. So I'll be giving you one research example, how I have used proteogenomics in my cancer research. So I explored high-grade glioma in my PhD. I did proteogenomic study. The project was in collaboration with Tata Hospital, where we recruited histopathologically and radiologically confirmed patients. And from those patients, we collected the tissues and we isolated DNA, performed whole genome sequencing. We isolated protein, performed proteomics, DIA-based proteomics. And very interesting thing that we found that the top mutation that we were seeing, let's say these were the top mutations, these top mutations were nowhere to be seen in the global databases. So TCG, I was mentioning, the Cancer Genome Atlas, they have huge database of Western population, and many times whenever we want to address a question about the cancer, we download those data sets. But the problem is we have very different like a lot of differences are there because of the genetics, because of the environmental factor, and using that databases are not actually the right thing, because their mutations and our mutations are different. So our top mutations that we were seeing in most of the patients were nowhere to be seen in TCGA data, or maybe very rare, like 0.02%. And the top mutation, the hallmark gene that were given by the TCGA, were actually from 52 to, let's say, the few, only the 13%, and then it's below 10%, right? So we need to have Indian population specific resources, right? And using Western databases blindly are not the right approach. This is what the genomics data tells us. We also performed proteomic study where we looked at the high-grade glioma and control samples. So proteomics nature were able to segregate the healthy samples from the high-grade glioma, right? You can see the control samples are completely different, forming different cluster with the from the high grade glioma. But we were more interested in these high grade glioma sample. You can see a lot of course they are separated from the control, but they are scattered, right? They're not forming a tight cluster, although they all belong to high grade glioma. That means a lot of subtypes are there within these high grade glioma, and we were interested in finding that. So you can see here. So WHO was telling us there are only two clusters, IDH wild type and IDH mutant, right? But we were able to identify a third cluster which are having overlapping. They were not belonging to IDH wild type and mutant. You can see here. Earlier, like according to WHO, there are only two classifications, wild type and mutant. But our subtyping based on the proteomic signatures that we have identified, we were able to find three subtypes within the high grade glioma, right? Not two. And of course, we explored the pathways, but those are not important right now because we were more interested in subtyping, right? So when we did our cluster based analysis, we were actually able to see a third cluster. So cluster 1 was mutant, cluster 2 was wild type, but we had third cluster, which is a mixed population. And then we were interested in looking at how this third cluster is actually, at the pathway level, what are the differences? So you can see a very stark difference between the C1 and C3, right? So they, like if we follow the WHO classification, this third cluster won't even exist. They will fall within these two, right? So we won't be able to understand these differences. So I will not be talking about the pathways now, but we were able to identify a lot of the pathways related to RNA processing, a lot of the pathways related to extracellular matrix, a lot of the pathways related to aerobic respiration, oxidative phosphorylation, which were showing very stark difference between these subtypes. So yeah, you can see here a lot of protein signatures were different within these three subtypes that we identify using proteomics data. And then we mapped our proteomics data with the genomics data. And we were what was the surprising thing is the cluster that we were seeing with from our proteomics data, we were also able to see within genomics data also. So we performed NMF clustering using genomics data, and you can see that these are the three clusters which and based on the IDH subtype, you can see here this is the wild type, this is the mutant, and this is the mutant, wild type, and the mixed population, right? And we were able to map our genomic cluster C1, C2, C3 with the proteomic cluster here, and a lot of mutations, subtype specific mutations we were able to see. For example, we were able to identify novel NCAD mutation that were C1 cluster specific. Similarly, ATXN3 and GPRN2 mutations we were able to map, which is cluster 2 specific, which is the wild type. And then the TCAN mutation we were able to identify. So all these three mutations are not reported by the TCGA, right? So these are first of all, these are Indian population specific, and these are mapping to different clusters. So that's how I used proteogenomics for subtyping, better classification to address the tumor heterogeneity, right? So cancer begins at the genome level, and but you need to understand what is happening at the protein level, and proteogenomics bridges the gap of genomics and proteomics. So the take-home message from my slides are genomics tells us what could happen, proteomics tells us what is happening, and proteogenomics tells us what truly matters in cancer. Thank you. You can follow us on LinkedIn for more updates.
Wonderful presentation. So I request all the participants to kindly ask your queries in the chat box. So there are some questions. So question from Praval. How can we create our customized database? Please mention some pipeline.
Okay. So for that, you need to first you need to have genomic information, right? So once you have those genomic information, then you can create uh, these databases. You can there are a lot of GitHub based pipelines which I will share, and you can use those pipelines to create. There are tools are available which can be used to create the databases, and then that database you can use to map the proteins.
Okay, so the next question is from Nomita. Hi, ma'am. As we know, there are many genetic mutation which is currently used for disease diagnostics in hospital. Is there any proteomics test available?
There are lots of, for example, for you are asking any particular cancer disease or in general.
So Nomita, can you open your mic and ask the question?
So I'll answer. It's generally like for the proteome, there are lots of proteomics based markers. For example, if you can see different kinds of protein. I can't remember for particular brain tumor. Brain tumor for the WHO, it's only the genomics they have included. Proteomics markers are not included in the brain tumor. But in other disease also, right now we only have genomics marker mostly. Protein level things we have not incorporated, but the tau protein is one marker which is known to have a role in neurodegenerative diseases. So again, I'm not sure if they have included that in a normal diagnosis, because that only a clinician can confirm. But through research, we know a lot of proteins which have a role to play in diseases, and tau protein in neurodegenerative diseases are very good example.
Okay. So Vipra Sharma raised her hand. Uh, can you just ask your question?
Hi, Ayushi. It was a nice presentation. I have a doubt related to that customized database only. So my question is, once we are analyzing our samples, so based on genes identified, we prepare that database and follow it up for proteomics integration and all,
Mhm.
or it is like that we usually do for proteomics. We take reference as from UniProt and do that way. So this is my confusion. Could you please clarify this?
Yeah. So that was the point I wanted to make, that if you're using a standard database from Uniprot, you might miss a lot of patient specific proteins, right? Because those standard databases are general databases, right? So you might not be able to capture the patient specific proteins. So for that case, you need to develop a patient specific database, and that you can only get if you have the genomic information available, right? So you get the genomic information either by whole genome sequencing or whole exome sequencing, and then you can, translate. So what is customized database here? So you are basically converting your FASTA file, right? What is Uniprot database? It's just the FASTA file. So you need to get the FASTA. So from your genomic, like WGS or WES, just create a FASTA file. And once that FASTA file is with you, that you can use that FASTA file to identify the proteins.
Yeah, so that's right. But still, suppose I'm using data of 50 patients. I have 50 samples. So,
Mhm.
patient-wise, it could have 10, 50 plus minus genes detected or difference in their mutation pattern. So, in that case, those data is plugged into one, or we take just random one
Hmm.
reference.
So it's
So,
up
this
to
is
you. Yeah, yeah, it's up to you. What we have done, in my study, I had 32 samples, hybrid glioma samples, right? So we also screen patient wise, like for every patient, we created customized database. Okay, so that way we had 32 customized databases. You can also create, like, let's say if you want to do population specific study, right? I want to only see what kind of signatures are present in Indian population. In that way, you can also combine your databases. Like you just use all your 32 databases and combine, and then you will have the signatures of all your 32. I'm just giving you an example, all 32 patients. So it's up to you. If you want to, like, for clinics, if you want to manage a particular patient in that way, do the patient specific thing. But if you want to do a research, population specific research, there you can combine and then report population specific signatures.
So follow-up question is, suppose I have used 32 patient samples in my genomic study.
Mhm.
So that should remain the same for proteomic study as well, or we can
Ideally,
take
yes.
another. Okay.
So here you want to perform genomics and proteomics on the same sample, right? So if you have taken, it's not about 32. It's like exactly same patient you are taking for, you are isolating DNA and you are isolating protein, and then doing the same thing, genomics and proteomics. So that's why,
Okay.
yeah, that is
Understood.
important.
And please do share that GitHub link so that we can understand the flow of doing that.
Actually, GitHub will give you the pipeline, but usually you need to just get the FASTA file and then you do the in silico translation and you have the protein. I can't remember the tools right now, but yeah, you can do that in silico.
Okay. Thank you.
Yeah.
So the next question is from Ronita. Great talk, doctor. Can you please tell me if there is a way where I can use AI in proteomics and genomics data analysis? And also a follow-up question is that I have a question to both Dr. Ucho and Dr. Verma. We are in masters from biotech. What do you think the AI era, the career will move for clinical research? What skills I should acquire? Most of the college don't teach these skills. How do you think, Dr. Ayushi, we can get this skill?
See, Uh,
There are a
what
lot
was
of
the
questions.
first question?
The first question is, can you please tell me if there
Huh.
is a way where I can use AI in proteomics and genomics
Huh.
data analysis?
Okay. So yes, you can use AI. So basically artificial intelligence or these machine learning models you can use to generate the classifiers. So very good thing that both the techniques, genomics and proteomics, will give you huge data, right? If you do whole genome sequencing, you will get thousands of mutations. Similarly, if you do proteomics, you will get thousands of proteins, right? So you have huge features. What you can do, you can learn AI tools or machine learning algorithms, and then you can use those tools to build a classifier model where you can, train. First, you of course train the model, and then you test your model and identify some of the good classifier which can, segregate maybe tumor versus healthy, or which can segregate different grades, different subtyping. So you can create classifier models, you can create algorithms which can, help you predict the disease. You can of course predict the disease based on the signatures present, and you can also subtype them, use those tools in subtyping or classification.
Okay, so the next follow-up question is for both Dr. Ucho and Dr. Verma. We are in masters from biotech. What do you think the AI era, the career will move for clinical research? What skills I should acquire?
I think everyone should learn programming, because of course the chat GPT Gemini is now there to give you the code, but you should also understand how these codes are getting generated. So you should learn programming languages. It will help you. So if you are to be in the omics field, or not just in the omics field, it will help you. So start learning the basic languages like R. Start with the R, then you can move to Python. But I think if you are comfortable with R, you will be able to solve a lot of things on your own. That is my answer. Dr. Utsu can answer.
Yeah, so it's mostly in the similar line. So there are not many courses, but yeah, there are frequent webinars like these, or boot camps like this. So one way is to participate in them and get an idea of what to look for, where to learn. And as Doctor was saying, though there are no code tools, I think there's some amount of familiarity with R and Python will be helpful. And also, since you are in biotech field, I think you have to know the tools like basic AI literacy is important, and also have the coding and tools like AlphaFold, how they help in understanding protein structure, understanding the deep variant AI models like deep variant. So there are multiple tools coming up every few weeks, few months. So we have to be staying ahead in the curve, know about them, which are relevant to your field of research or work, and try them out. YouTube is always there. You will find somewhere somebody teaching some of the basics in YouTube if there is no webinar also. And obviously there will be courses from Swastome also. There will be courses from others also. So that's it from my end.
So yes, the last question is, college don't teach these skills. So I want to address this question. You need to just outsource this kind of courses, like from Swastam or other sources. You need to outsourcing these courses from YouTube or other online websites. So, Vipra, do you have any question?
Yeah, I have one more question for Ayushi. Uh, uh, I want to know that data you shared with us, it is AI based or statistical analysis based?
That is all statistical analysis. No AI tool was used in analyzing my proteomics data. So I've used for genomics, we have used Galaxy pipeline, and for proteomics, we have used Perseus and standard genomics and proteomics tools for analysis.
And for data visualization, it is through R or something else?
Usually, the Python was used to create the heat maps.
Okay, thank you. And one more thing for that slide number 13, if I remember, there is something looking like networking with branches and all. What does it represented actually?
I don't know which slide you were talking about.
30 number where there is a branches and networking like thing.
Those were the kinome map. Those were the kinome map.
Okay,
Uh, yeah, kinome map. You can search on Google, you'll get it.
and that is also prepared using Python.
No, no, that was generated using the kinome tool. So,
Okay,
yeah.
yeah, thank you.
So I think there is no more questions. For Mansi, the mind meet will happen next month. Uh, so I is there any question from any participants? So I think there is no more questions. So as we come to the end of today's session, I would like to sincerely thank our esteemed speakers for their valuable time and insights. On behalf of Swastome, Jivomics, and the Vigyan Chronicles, thank you all for being a part of MindMeet. We will look forward to seeing you in our upcoming sessions. Thank you everyone for joining today's session.